pleasure for me and great honor for me to introduce him to you. And of course, everybody in this audience knows quite well that Jim discovered the structure of DNA, the famous double helix. And he and Francis Crick were awarded with Nobel Prize for this discovery. However, many of you don't realize that this was not just a discovery. This was the beginning of the new modern biology. That biology, what exists today. So all modern biology came from double helix. And moreover, not only modern biology, but also the future medicine also depends on this discovery. So after discovery of double helix, Jim moved to Harvard, United States. Double helix was in Cambridge, England. And there he had a very active department with many brilliant scientists like Mark Taschen and Wally Gilbert. And there were a series of very important researches developed under supervision of Jim, mostly devoted to the control of realization of genetic information in cells. Then Jim moved to Cold Spring Harbor as a director of Cold Spring Harbor Laboratory. And under him, this laboratory very soon converts to one of the major, most important centers of molecular biology throughout the whole world. The Cold Spring Harbor Symposia, which I visited pretty often, were one of the central events in the molecular biology in those days and also now. In Cold Spring Harbor, Jim was mostly interested by cancer problem. And the collective which he combined there made a lot of very important works in understanding how the normal cells is converted into the cancer cell. And Jim was actually one of the activators of all this program, big program. I wish to say that Jim is characterized by great scientific provision. He was the initiator of the program, so-called Human Genome Project. Actually, it was a determination of the base sequence of the whole genome of human. And many scientists at that time, many very important scientists, did not believe this project is realistic. They believe that it's completely non-realistic project. But Jim was, as always, right, and the sequence of human genome was solved even sooner than it was planned at the beginning. So it was, again, very important achievement of science in general. And now, as I understand, the main interest of Jim is concentrated in brain and different kinds of brain diseases. And I hope that today we will learn a lot about this very important question. So, please.
No, I don't need. Okay. Yeah. So I want to thank my hosts in Moscow for giving me really a very interesting and rewarding week in Moscow. It's much better than even I hoped. And now I want to particularly thank this House of Sciences for giving the opportunity of such a large audience. And those who are running the House, I want to thank them. I'll speak a little, to start with, about my philosophy of, how to get science done. And because you can sort of I'm just 80 years old, and so for 20 years, or, once I got to school, for 20 years I was a student. And then for 20 years, I was an active scientist. First doing DNA, and then at Harvard University having students working on protein synthesis. And then at the age of 40, I changed sort of the course of my life, is that I became the director of a scientific institute interested in genetics. And there were several reasons. The laboratory had no money and would fail, but probably the chief reason was that I wanted to start a rather big effort of how to understand how viruses cause cancer. And I could see that as a project much bigger than I could do in the university. And so my interest would be best served by being the boss and hiring other people to do science. So after the age of almost 40, I didn't have any more students. I didn't have a laboratory. So in becoming the director of the institute, I had no laboratory. So my chief function was just to hire people, get the right people. And if you have a scientific institute, I think they're perfect devices for working on a problem. Whereas universities are not very good places to, say, understand cancer. You can do it, but you have all sorts of colleagues, you have competing interests, and it's hard to really focus in a university. If you want to focus, a scientific institute is the way, and I wanted to focus. From the start, I wanted to have the institute populated by young people who just, the bosses were people who had been postdocs before I hired them. So there were I didn't try and hire big people. I tried to hire people that no one had heard of, but who, for some reason, I thought would be good. And of course, the chief reason that you wanted to hire them is they wanted to work on cancer. It wasn't they wanted a job. They wanted to understand cancer. My job was to find the money. So, I could find the money, and then once they were hired, I really didn't ever tell them what to do. And my rule for sort of best running it is delegate as much power to other people. Try and have all decisions made by other people if you can. So I had an assistant director. So he made a lot of decisions that saved me a lot of work. And I tried to make myself so unnecessary that I could go to Europe for a year and it wouldn't make any difference. because I wasn't really telling people what science to do. I hired other people for them to decide what science was doing. Now, initially, our institution was very poor, and so there were no permanent scientists. In fact, that was a very good thing because I didn't want anyone to stay on very long. I just wanted them to do good science, and knowing they had no security, would never get tenure, would then move on to a good university. So I could keep the age of people under 40 by not offering security. Because when I had got into science, at the age of 20, I came into the world of Max Delbruck, the physicist, German physicist. And Max was always constantly telling me that beyond 25, you begin to go downhill. So by 40, you're... and so... and now, so that was the policy. No security. People work hard, do science. They do the science of their own ideas, and then they find a job some other place. Okay, so I was effectively the boss for about 25 years. And ran that way, and we became successful essentially because I was never bored by the science done at the laboratory. Everything people were doing, I was interested in. That's why I hired them. And second, my job was not to bore the people who would give us money. So I had to actually be excited by what we were doing, and the laboratory luckily was located in a region of some very rich people, and through these very rich people, we could move much faster than we could if we were totally dependent on the government. So good science is sort of essentially elitist. You're trying to move faster than other people, and to be different from other people, you have to, it pays, at least in the United States, helps to have some private money to let you to move faster. So I never really worried about money for 25 years. Because the science was good, the neighbors liked us. And but we kept getting bigger and bigger. And another thing besides reserving the power, of who we hired, I had all the power of what buildings would look like and what trees we would plant. So it's been my feeling that scientists are not, on the average, very interested in aesthetics. They don't care what things look like as long as they can do their experiment. But if you want an institution to have a long future, you probably want it to have some grandeur. And if it looks slightly grand, people are more secure giving you money because it doesn't look like you're going to fail. So my job was, I got a lot of pleasure from trying to design handsome buildings that looked like they would stay permanent. And planting trees, now I see some trees that I planted 40 years ago and they're getting big. And my successor has not followed the way I did it. He's let people get old. And sometimes that pays sense. Actually, after I'd been there in Cold Spring Harbor 15 years, I gave security to someone. So he got tenure. And it's about basically his work I'm talking now. He's 57 and he's still acts young. So you can have rules, and then you should break them. But it's hard for universities to break rules because it seems unfair to someone. So it's tricky, you know, to just improvise. And the other thing, which is I think why I was successful as a sort of boss, is that I wanted things to happen as fast as possible. So I would always make a decision as soon as I could. Someone would come into my office and I would give him an answer, not saying I will wait a week and think about it. Just say yes. I found it was a very good policy to say yes every time someone came into the office because it showed your faith in them. And, you know, you should say no, you should fire them. So you want people that you can say yes to. So I never really had appointments. My door was always open. And I tried to move fast. So one way to move fast is to have no committees. Just no committees. Just make decisions. So we never had committees to hire people. We just hired people. Now we have committees. It slows you down because you try and be fair to everyone. Whereas I've found that in science, it doesn't pay to be fair to everyone because it just slows you down and, you know, if you don't do something, someone else will do it before you. So, and then the other is I thought if we do something, we should try and be at least as good as anyone else in the world. Never do something unless you have a chance of being at the very top. Never go in and say, well, we'll be number 10. Either you go in and think you'll be 1, and of course you're happy if you're 3. But at least so, initially, you know, our competitors, well, once initially are not competitor, you know, people in London, and they grew old, so we didn't think about them. And for a long time, just basically, I've tried to be as good as MIT, with much less money, and with superficially much less distinguished people. So MIT can afford people who, you know, everyone else says is good, whereas I had to hire people that before anyone else said they were good. Yeah. So I probably didn't, if you think about it, having my own lab, doing my own science would have kept me from thinking about my competitors. So I always think because that we want to be first. That's all. And sometimes you are and sometimes you aren't, but at least you should have the objective. Okay. So now I'll give a talk on through the work of Michael Wigler, who developed a technology for finding genetic differences in cancer cells before you could sequence them. So, and I will explain it in a moment, but Wigler is an unusual person. He would make a terrible boss. Because he really only thinks really about himself, but that's very, by himself, by his own science. And what Mike always needs is someone like me who supports him. But before I hired him, he had been a very successful PhD student at Columbia University Medical School, and had worked out a method for getting DNA into cells. His aim was to make a normal cell a cancer cell by adding DNA. And this way you can isolate a cancer gene. So it was a technology which he developed, and then it turned out to be very useful commercially. In getting the genes into animal cells to make biotechnology drugs, proteins. So Wigler's work led Columbia University to get a patent which made Columbia University $600 million. So Wigler himself collected about $100 million. So Mike is a rich scientist. But after becoming rich, his science has gone on at the same high level. So he never, wanted to be a scientist to become rich, or, to do it. Being rich just let him have more freedom. So he lives in a big house across the water from me, which most people will know was the summer house of John Foster Dulles. Which, when you think of this, it makes me laugh. That scientists can be as successful as Wall Street lawyers. Okay, so I will then. Say, so these are some of the sort of rules. And a sort of rule that is a really difficult rule. I'll just go back to that. An institution is either getting bigger or smaller. And so if it's successful, it will get bigger. But you can't get bigger forever, so that's a problem. But if you don't build new buildings, your good people will go some other place. So success demands growth. And that's sort of true of all companies. Companies have to get bigger, and sometimes they collapse. You get too big. So just to conclude, what is the solution to Cold Spring Harbor getting too big? And so I want to turn it into a university. More or less to keep it alive. If we don't become a university, then we will collapse. And that means giving people tenure. And sort of my rule is I really only want to give tenure to people over 50. Not at 35. Because if you can go through to 50, you probably won't burn out. Okay. Okay, here's Wigler on the left, and the group. So his group is about 15 people. So it's not 40 people or something, it's about 15. And about half of them are trained in mathematics, because when you work at the level of the human genome, you have to be trained in mathematics to analyze the data. And Wigler, when he was a student at the university, was a math major. So Wigler likes math, okay? And the tree behind you can't see, but it's about 120 years old and a beautiful tree. Okay, so Wigler's method was that with the human genome, you had the possibility of isolating regions where a gene normally present in 2 copies suddenly would go to 3 copies. Or you might lose 1 gene, and instead of having 2, 1. And so they published a paper, and it was now 4 years ago. The first paper saying that if you look for the genome not at the level of chromosomes, but use DNA chips, copy number polymorphisms, every person contained about 10 big ones, because the first technology only picked up big ones, okay? And then the work was confirmed by someone else. So until this work, everyone studied simple mutations, changing one base to another. But this was focusing on these, and so, and this is out of date, but I think the interesting thing is that about half the genetic variation in humans is caused by these changes in gene number, and the other half of changes in human is due to A turning to T, or adding a base or losing a base. But some of these changes can be 1,000,000 bases in length. They can be quite big and contain many genes. So, yeah. And something is not showing up. Oh, okay, yeah, I did, yeah, okay. So this is really what, in a simplification, the human genome, all human genomes look like, is that along all the chromosomes, you find regions where the copy number is either increased or decreased. And the reason these occur, and I speak now to experts, is because of repetitive DNA, you get none crossing over, illegitimate crossing over. So the regions, the enzymes which are trying to do crossing over, think 2 regions are the same, but they are the same, but they're located on different parts of the chromosome. And when you get this crossing over, so one can say, given the human genome, there's copy number variation will always occur. In fact, it will occur pretty commonly. Okay, so what Wigler first essentially tried to do was to see whether if you had a case of, this is studying autism, the disease where the children cannot speak and where they have great, in some cases, extreme learning problems. Their IQ is low, and then their behavior is often repetitive. They're extremely frustrated. And it's not surprising that their behavior. So as the human diseases go, the worst thing that can happen to parents is to have an autistic child. I mean, it's very, very difficult to raise a child. Okay. So what Wigler did was to look at DNA from when both parents seemed normal and the child was autistic. And what they found was that about 10% of the autistic children lost rather big sections of DNA. So autism was caused by losing DNA, having 1 copy instead of 2, but it could often be a region that would contain 20 genes. Then Autism before Wigler was thought of possibly of being only happening when several different genes made your child autistic. But it's a funny disease because you get a deletion, and if the deleted chromosome becomes part of a boy, he becomes autistic. If the chromosome is in a girl, the girl will not be autistic. So for some reason, the ratio is about 5 to 1. So some girls get autism, but many fewer than boys. So if you get a girl and she inherits an autistic chromosome, she will, she can talk and all that, but half her sons will be autistic because you have the bad chromosome. And if it goes to a boy, so that essentially the chromosome will be lost in a couple of generations. It will appear in boys and then it will be gone. So autism, as you just seen, is not due to pre-existing genes, but the creation of new genes. Now, there was a theory which was proposed by a psychologist at the University of Chicago just after World War II, and he had a school for autistic children because parents couldn't handle them. And he observed that the mothers of autistic children, he said, had very little interest in their children. And he called them iceberg mothers. And so the cause of autism under that sort of was that the child became autistic because there was no affection shown by the parent. And then people didn't like that. It sounds terrible. But probably the observation which has been repeated by the English psychologist Simon Baron-Cohen, first in London and now in Cambridge, is that the women who are carriers of an autistic chromosome are slightly autistic and have no interest in children, or very little interest in children. So it sort of looks like half of autism occurs to just out of the blue. So the mothers have affection for children and they're not different from anyone else. But if they've inherited an autistic chromosome, that leads to lack of affection toward children. So why women are protected? Being protected is slightly a curse because then you're children. So it's not nothing nice about this result. What people are now trying to do is to make mouse models of autism, take genes. The trouble is that the cases, well, let's just show you one. Well, that most of autism will probably be due to a mutation which is just new and isn't the same as a pre-existing change. Well, let me go on to the second. Oh, and you can just see, now there's a second complication, or a real complication of autism, is there was a condition called Asperger's syndrome, where the children have normal intelligence but very poor social interactions. And now that's called a type of autism. And it's more common than conventional autism. And so the frequency of autism, which used to be said was 1 in 1,000 children, it gets close to 1 in 100 if you include Asperger's. So these are people who are obsessive, don't interact well with other people, but can be sometimes extremely bright. And the ratio of boys to girls is 10 to 1. People have written that Bill Gates is Asperger's. So, you know, he enjoyed computers as opposed to people. So, whether it's true. And whether it's right or wrong, some people now are writing that Einstein was possibly a little autistic. And it may be true that half of the really talented mathematicians are somewhat Asperger's. So there's a connection between Asperger's syndrome and mathematics, which will be very interesting to work out. Because some mathematicians are very good with people, but even when I was a boy, I was told that mathematicians were strange. So I think there's probably some truth to it. The most extreme are the idiot savants who can do these fantastic calculations and who virtually are hopeless by themselves. They're idiot, you know, you call them. So to me, it's possible that there's been a selection against mathematical ability in many societies because it makes you worse with people. So it's we're just at the beginning of studying it. Okay. Yeah, these are just two kind of, and initially we thought these copy number were about 10% of autism, but now we're with the DNA chip technology lets you look at smaller and smaller copy number variations, and it's certainly going to be at least 30%. So, and maybe 50% if the small ones are harder to study. So half of sort of human genetic disease may be due to these sort of changes, and the other half of the more conventional type where one base changes into another. But if you want to work at the genomic level, DNA sequencing has been until recently too expensive. But a big change is that we'll soon, through copy number variation, we will discover regions of DNA which, if they're removed or too much of, lead to autism. And then we will sequence large numbers of autistic people to see how much of autism is caused by conventional mutations. So that 10% we may go down to 50% as the technology gets better. Okay. Yeah, okay. That's where sort of think we're going. And a lot may be SNPs that haven't been discovered. So we'll see. So many of autism, the heritable ones are when the gene is first inserted into a woman. Then it can be inherited. Okay, so then I want to say a little about schizophrenia. So where conventional genetics was pretty disappointing. And there were initial hopes that schizophrenia might be caused by only a few changes. But if I can give the conclusion, we think schizophrenia will be much like autism, and a very large number of different changes in different genes will upset the functioning of the nervous system, so you either end up in autism or schizophrenia. But I should emphasize we are just at the very, very beginning. So almost everything I say could be wrong, except I don't think so. Okay. So this is essentially what you do. And so we're beginning to see copy number variation in schizophrenia, but in a large number of different cases, different genes. So, you know, you could say now, if you combine, and I should say this is the sum of MIT and our own lab. So even though we're, in a sense, competing with MIT, we're talking to them all the time. So we share the information, okay? And the probability values are too low. I mean, are not good enough yet. So it's our impression we'll probably have to look at about 30,000 people with schizophrenia before we get an idea of the universe of changes. And so it costs right now about $1,000 per person to look, if the DNA has been collected for some other reason, just our work. And so 30,000 is about $30 million. So the sums of money are large. And so I should say, where did we get the money to spend millions of dollars? And it didn't come from the government. It came from parents who had autistic or mentally ill children. So all the work on autism is supported by a foundation set up by the mathematician Jim Simon, who gave up being head of the math department at Stony Brook to become a financial investor. And last year, he himself made $3 billion through mathematics. But he has an autistic daughter, okay? So he supports that work, and there's another family called Stanley who have a bipolar son, and they've given already or pledged about $200 million to study schizophrenia and bipolar disease. Now, one thing one is discovering now is that we're finding genetic regions where if you get changes, sometimes it leads to autism and sometimes to schizophrenia. And we don't understand why. In other cases, you know, it's a spectrum of, let's see, here. Yeah, these were, yeah. So this child was called, you know, normal, but it had attention deficit disorder. So it really looks like it's a dominant gene, or the effect is. So it's going to be complicated in that you'll get a change to the nervous system, which, depending on your other exact genetic composition, could lead to any depression or so on. Okay, and this is just evidence for those of you who are experts of what the changes look like. So this was region 22 is a very, on chromosome 22, is one that's been long known to be unstable. And this is neuroregulin. So Asperger's, schizophrenia. And then they to sort of finally summarize that we've only looked at about, you can see 200 cases. And in people with schizophrenia, we're observing copy number changes which we've never seen before, that you don't see in normal people. And they largely occur in genes which control the functioning of the nervous system. So the frequency of schizophrenia is generally said to be the same world around, 1%. And we don't know how long a schizophrenia, and it's been debated, are the changes dominant or recessive? And I won't go into the reason, but it increasingly looks like they're at least semi-dominant. So that if one of the parents has schizophrenia, half the children will have some abnormality, though in many cases it will not be schizophrenia. But the child will be not quite normal. Schizophrenia has the really horror of the disease in opposition to bipolar disease. It's accompanied by a lowering of IQ by about 15 points. And that this IQ difference goes back to early childhood. So from early childhood, their nervous system is not working well. And it's not just conventional learning. Children who are going to be schizophrenic walk 4 months later. So walking is you have to learn. So these are, it won't be true of everyone. There'll be some, since so many genes are involved, I'm oversimplifying. But there's, so schizophrenia consists of first a weakening of cognition and learning, which in later life, generally after adolescence or during adolescence, can lead to psychosis. The drugs which are used to treat schizophrenia control the psychosis and have no effect on cognition. So most victims of schizophrenia, so it's almost as if schizophrenia is a people of disease, and there are exceptions, but having a low IQ increases the probability of having schizophrenia. Whereas bipolar disease seems to be, if anything, the mania, if it's not accompanied by depressive psychosis, can make you more productive. So the message, I guess, I'll just conclude, is now the genomic techniques have proven without a doubt that schizophrenia has a genetic component. It's going to be very complex, and we will be lucky if we really have the main picture clear in 5 years, but certainly in 10. And once the genes can be isolated, then we will put these mutant genes in mice and see if you could cure mice. So they're already, mice don't get schizophrenia, but when you put the gene in, they behave slightly, their social interactions decrease. So there's something in common. So I think it's going to be a very exciting one. You can say, well, why am I so focused on schizophrenia? Well, it's very simple. About the time we were thinking of starting the Human Genome Project, we began to worry that my eldest son was schizophrenic, and he is. So, it was a realization that we were unlikely ever to understand the disease until we really got the human genome, and then until we got the technologies that reduce the cost. We would never be able to do it. I have sort of hopes that within 5 years we'll know enough so I could look at my son and find out what's wrong. Doesn't mean I'll be able to cure him, but at least we will know what's wrong. So it's now is the time really to study the genetics of psychiatric diseases, and the real way is, getting family history and all that, plus DNA. And just to say something about the cost of doing a complete genome, my genome was done for, let's say, a cost of $1 million. That was 18 months ago. Recently, 2 people have been sequenced for using a different technology, a cheaper one, for $100,000. And so probably within 5 years, for the cost of a cheap American or Russian car, you could get your whole DNA sequence. I don't advise people getting sequenced as opposed to buying the car. But, if your son is or daughter isn't behaving normally, maybe then you should look at the DNA. I don't think you want to look at the DNA and predict that someone will be schizophrenic. I think we're so far from that level of knowledge. But it's very satisfying to me to think that if I can live another 10 years, then we will understand schizophrenia. And to end it, people who work on schizophrenia over the next few years will not be boring. So, they will be doing something and should be excited. So, that's my message, and again, I say how happy I am to be back again, seeing my friends in Moscow.
У нас есть время для вопросов, и в помощь нам в этом процессе я хочу пригласить на сцену Николая Казимировича Янковского, члена-корреспондента РАН и директора Института общей генетики РАН.
Как известно, около 200 тысяч белков, открытых ныне, определяют взаимодействие организма с окружающим миром и внутренние процессы. И примерно 1,5 или 2% наследственного аппарата кодируют эти 200 тысяч белков. Как, на ваш взгляд, для чего природа заложила в наследственный аппарат человека такой огромный ресурс, который пока не реализуется? Спасибо.
Yeah, yeah, I know. Yeah. The question is, why do we have more proteins than we have genes? And the answer is a phenomena of RNA splicing, whereas the original RNA molecules are processed in slightly different ways, yielding different numbers of proteins. And in some cases, there's the potential for an extraordinary number of different proteins. How important this will be, I do not know, but some cases some mutations are going to affect splicing, and protein defects will be the cause of the disease, but I can't be more specific.
I think I will ask in English because it would be just faster. I want to ask not about science, but about funding of science. What do you think, is it okay, the current situation with science, that most scientists, after they have their PhD, they need to go to biotechnology companies because the salaries are incompatible? And in fact, the Genome project was finished only with the help of Celera Genomics and other biotechnology companies. And is it okay? What do you think about the current way of science? I think it shouldn't be changed or not.
I think the way of science is very different when I was young, because there were very few scientists, and there was the amount of money per scientist was much greater because there were fewer numbers. But at that time, the only jobs were academic jobs. I think we probably would expect that many people trained with PhDs will go and work in industry or become journalists or become government. And I think will be a very good thing because at least in the United States, we don't have enough technical competence in our government. So I think we really have to sort of expect that we're training people sort of to show them the scientific method, how you do it, but then you'll go out and your career will not necessarily have any connection to what your PhD thesis is about. That's both good and bad. But I think, at least in the United States, we have a shortage of well-trained people to run our country. On the general thing, the salaries of scientists is far too low. And I'm sure right now people in Russia who would have become scientists now want to become businessmen because they can buy a better car or have a car at all. On the other hand, that is not necessarily a bad thing for Russia right now because you had to get your business going to generate the money to pay for the science. So having bright people go into business is not a bad idea.
But bright people they are not like bright people.
Спасибо за вопрос.
Okay.
Еще вопросы?
Здравствуйте. Скажите, пожалуйста, а если исследовать не только болезни, какие еще перспективы может дать изучение работы генов, отвечающих за функции головного мозга?
Yes, it's whether the genome is only useful for disease. I think it will eventually tell us a great deal about human personality, our emotions, and so it won't just be disease, but just what makes us human in an interesting way. I've always viewed human beings as products of evolution. And so through studying the human DNA, you will better understand what human beings are and what motivates people. Then there's, biotechnology, which Russia has been, like other European countries, and not given enough emphasis to. It's been America and China now. I think there's enormous use of genomic DNA methods in producing useful commercial products. And hopefully we will get new sources of energy through better, getting better enzymes, just trying to improve human life. So I think people who really become experts at DNA should, if they also have common sense, will succeed in life.
Dr. Watson. Professor Watson, could you please comment on the work of the well-known psychologist Arthur Jensen, who claimed that the human intelligence has an inheritable genetic component, and about how this is related probably to the recent scandal in which you were involved personally.
Well,
Thanks.
I never would intentionally have made that statement, which greatly embarrassed me, and I apologize for. The general statement of Jensen that human intelligence has a genetic component is a total fact. There's no doubt about that. It's really the variation between different people, the origin of nature versus nurture, but that it's not at all surprising that our abilities are a function of genes. If our brain works very well, we will be more successful. And unfortunately, there seems to be a difference of abilities is at least shown by tests. What those tests mean deeply, I don't want to say. But I don't think it's unfair to say that Arthur Jensen was an evil man. He was treated as an evil person, but he was not. He was a very, very good psychologist.
У меня просьба к организаторам, которые передают микрофоны, смотреть в первую очередь на нашего гостя или на меня, чтобы видеть, кому микрофон надо передать. Пока, Ирина Геннадьевна.
Доктор Уотсон, при изучении таких вот умственных расстройств, о которых вы говорили, возникает масса этических проблем. И у меня вопрос следующий. Каков ваш прогноз, по какому пути пойдет общество, когда будет уже известно, так сказать, изучены все гены, влияющие на развитие шизофрении и аутизма, по пути абортов тех плодов, которые могут в последующем развить эти заболевания, или по пути все-таки лечения их?
I want to start with this. Genetics didn't create the ethical problems. We have life today without genetics is filled with ethical things. Some people succeed, some don't succeed. How do you prevent too growing inequality in society? I think we will, through the knowledge of the genome, we will at least know what the true nature of the problems that society faces. And I think we will treat each other better. As an example, we sort of blame mathematicians because they're not more social. Well, maybe they can't be, so we shouldn't blame them. We should just be very lucky that they're good mathematicians, instead of saying, they're not going to be good on a television program.
Доктор Уотсон, у меня вопрос следующий. Я хотел спросить, исследовались ли эти дупликации, о которых вы говорили в своем докладе, у других приматов? И в связи с этим следующий вопрос. Могут ли эти дупликации играть роль в видообразовании, скажем, появления человека как вида?
Well, if you look at chromosomes of different species, they're often quite different. And that shows there's a lot of evolution at the level of these chromosomal changes. So the changes I've been talking about are very important, will probably turn out to be very important factors in evolution. Even though most are harmful, some will be beneficial.
Скажите, пожалуйста, как вы считаете, могут ли через 10 лет или через 50, допустим, в школах по генетическому анализу определять способность детей, допустим, к математике или к музыке, или к занятиям литературой, или к занятиям биологии, или это невозможно технически? Или, может быть, это и не врожденные способности, а все зависит от воспитания? Спасибо.
It's a very difficult question to ask, to what extent you should send children with different ability to different schools. It certainly works for music. And so you want to let children work at their abilities, and some children have many more abilities and should be allowed to move faster. So I don't think you want to put everyone in the same school. I was very lucky that I was taken and put into, you could say, a very elitist school, which trained me much better and was sort of compatible with my own desire for learning.
Скажите, пожалуйста, известно, что психопатология проявляется при разных метаболических расстройствах, с одной стороны. А с другой стороны, очень много генов, которые приписывают к шизофрении, и они все очень с маленьким эффектом. Может ли быть так, что каждому гену соответствует какое-то метаболическое расстройство, и это объединяется в шизофрению по формальному признаку, что такой отдельной болезни нет? Может быть так?
Well, I think our personalities are largely dictated by our genes, not by the way we've been raised. You're sort of born with your personality. What genes will, it'll be fun to study people who are exceptionally happy. We'll probably learn something. Yeah. So I'm basically allowing, I think people should be allowed to satisfy their curiosity by trying to find a genetic origin to human characteristics. Forbidding a search for finding these things, I think, would be, a mark of a fascistic type government that really doesn't want you to be free.
Профессор Уотсон, у меня вопрос, прошу прощения, не совсем по мозгу, скорее по структуре непосредственно ДНК. Вот открытая двухспиральная структура, она очень хорошо описана сейчас, посчитаны углы между разными группами атомов, и в принципе это преподается и подается как неотъемлемое химическое свойство самой молекулы, именно такая конформация. Однако я недавно услышал, что сейчас теория разрабатывается о том, что такая структура это не есть химическое свойство, а наоборот поддерживается ферментами. И то, что ДНК вполне устойчиво, возможно, чуть менее устойчиво, но все равно вполне себе жизнеспособно, может существовать и в очень многих других конформациях.
You're talking about, are there changes other than changes in the four letters? Sort of epigenetics, chemically modifying the DNA by modifying the proteins that are around it or methylating. That's a very hot topic in biology. And I can't give you an answer of how important it'd be. We know that some of these changes seem to be involved in cancer, but it's not clear whether they're the majority or even just a tiny minority. We don't know. That genes are not the total answer is shown by the fact that identical twins are not completely identical. Yes.
Профессор Уотсон, известно, что вы в ваших широчайших исследованиях занимались и проблемой расовых отличий ДНК, что вызвало ажиотаж в кругах расистов. Вот. К сожалению, такие люди есть и в России. И ваше слово в этом отношении – это слово авторитета высочайшего значения. Можете ли вы сказать вот людям такого рода, что все расы все-таки равноправны и человечество едино?
No, I think, are there genetic differences between people in different parts of the world? Yes. All I want to say is that when you have evolved in separate places under separate environments, you should not expect the genes to be the same. Whether you call it equal or unequal, but my skin is too light, so I get a lot of skin cancer. So, other people have darker skin. So, to say that everyone in the world is essentially the same, that's not true. My son isn't the same. And mutations can be very devastating. The belief that evolution has stopped, say, 10,000 years ago, that's probably just nonsense. It's just going on. If you separate populations, they're going to go in different directions. I think the future will give the answer. I never wanted to be quoted in a newspaper about IQ because I'm not an authority on it. I couldn't defend any statement I made, so I was very embarrassed that I seemingly insulted people that I did not wish to insult.
Professor Watson.
Просто.
Mr. Watson, did you know something new or about something interesting about yourself from the sequence annotation of your individual genome?
Could you repeat that? It came so fast I didn't hear it.
Oh, okay. Did you know something new or something interesting about yourself from the sequence and annotation of your individual genome?
My own sequence, it's 3 billion letters, so I've never looked at it. So I'm relying on other people to tell me if it's, until you sequence thousands of people. The only thing my sequence has told me why my stomach aches when I eat ice cream, because I only have one copy of the good lactase gene. So milk is bad for me. So that my genome told me. But otherwise, it has not changed my life at all. And I never expected that it would probably change. What I thought is maybe I would get some information which would benefit my children. That I might be carriers for some genetic, have one copy of a recessive disease gene. But no, we haven't found that. So I have no advice to my children. For a brief while, they told me I carried the breast cancer gene, which would then have a probability of being present in my nieces. But it turned out that the change which they have, they say it shouldn't cause breast cancer. So, I haven't lost any sleep. I had told people I did not want to know the structure of the gene for the protein apolipoprotein E, which is slightly prognostic for Alzheimer's. I did not want to know whether I was at risk to get Alzheimer's because I can't do anything about it. but I've reached 80 without Alzheimer's, so I sort of figure I'll reach 82. But you know, you can't really say. I have a grandmother who died of Alzheimer's, so I have 1 in 4 probability of carrying the gene that made her sick. But you know, so I don't think people should. Craig Venter, he released his own genome, and he has 1 bad copy of the Alzheimer's promoting allele. So unless we can cure Alzheimer's, Venter may be at risk, and 10 years from now he may regret knowing that. But you know, time will tell. Last question.
Профессор Уотсон, будьте добры, скажите, пожалуйста, в коей мере ваши исследования подтверждают или опровергают философско-эволюционные обобщения Ричарда Докинса?
You know, about Richard Dawkins, he's a very clever, intelligent person, and his book, The Intelligent Gene, was a very provocative and I think correct book. Dawkins has written many books, and his latest is about religion, which I think it's best that I say nothing about. Okay.
Thank you very much, Dr. Watson.